Lesson 4.5 Learning Goal. I will be able to describe how biotechnology edits DNA to solve problems and evaluate benefits and risk. The big idea is biotechnology is when humans use DNA science to solve problems such as fixing people who are sick with medicine, providing more food for a population, or conserving species that may have passed away or are on the verge of being extinct. Let's go over the key Regents terms that will help you be successful. Starting with biotechnology and genetic engineering. This is using DNA or living things to solve problems. So they use something called a restriction enzyme that can cut and paste together DNA from different species to make it more helpful. Gene therapy is treating a disease by changing a person's DNA in their cells. They often change the body cells, not the sperm or the egg cells. They do this by inserting a healthy gene to replace a faulty one or a gene that's not working well. CRISPR is a gene editing tool. This tool can cut DNA at a specific location so it can be changed or edited and a better gene being put in place. So let's take a look at a Regents level case study. CRISPR and fixing sickle cell. So in some people, red blood cells become a sickled shape, which can block blood flow and cause pain and a lot of organ damage. But in some regions of the world, malaria is very common, and we find a lot of sickle cell alleles at a very high frequency. So let's kind of look at the solution of how scientists came up with CRISPR for fixing this sickle cell trait. First, they gather stem cells from a patient, and stem cells are cells without a specialized job yet. So they're just kind of waiting to see what type of cells they will become. Next, they use those cells and the CRISPR edits the damaged DNA. The switch is then made to the gene that turns it from being damaged to producing the blood protein shape that is correct. So the blood protein shape changes, and that means you will then have a working blood cell. So our question is, how can the same mutation be harmful for some people, but helpful in certain populations in certain environments? So first we have hemoglobin, and this is a protein that carries oxygen in red blood cells. Having malaria is a selective advantage in regions such as South America or Africa because malaria is a disease that is caused by mosquitoes. And having this sickle blood cell shape makes mosquitoes less likely to bite you in these areas. So the disadvantage to having the sickle cell trait is that it causes pain and organ damage. But the advantages is that you'll be less likely to contact malaria, which is a severe disease that is also very uncomfortable. So overall, this is a mutation that changes your DNA code. So beginning with, you have a DNA mutation that creates a sickle allele. Some people have only one of the sickle allele, and that can make it better for survival in areas where there's malaria highly present in the mosquitoes. That means that because this is a beneficial trait, even though it can be a little harmful for the body, that the sickle allele or the traits stay in common in these areas.